site that is referred to as the operator. Remember, in the beginning of this lecture, I is a position that functions like a switch. But how does it function like a switch? It functions as a switch, for example, a turn-off switch, when it is bound to a repressor molecule. So in this case, the repressor molecule first has to be synthesized and then move, or should I say diffuse, in the cell to bind to the operator molecule. What it means is that the operator site has a DNA binding site for this repressor molecule. When this repressor molecule is bound to the operator, even if RNA polymerase binds to the promoter, these structural genes cannot be synthesized. There will be no transcription, there will be no translation. What that means is that these genes, there's no lactose in this environment, okay? And the cell does not, these genes are essential for lactose utilization. So normally this is going to happen because there's no lactose in the environment. So why should these genes be transcribed? So one way to regulate that transcription is to produce a repressor molecule that will bind to the operator to ensure that the transcription of these genes is blocked. So this is an example of negative regulation. Why? The product of the repressor gene has produced a molecule that binds the operator and blocks transcription, like repressing transcription. These are notes that you can look at and expand in your own time. Now, I want us to find out this operator, once it is bound to a repressor, can it be removed from that point to allow for the transcription of the structural gene? So the question I'm trying to answer is, how does transcription start? How does transcription start? This thing should be removed when a repressor blocks the operator. How is transcription of the structural genes going to start when at this point we have the repressor molecule bound to the operator? So in this case, the gene is turned off. So how is it turned on? So this is our current state. Within the inducer, should I say, this is our current state where we have our repressor gene synthesizing our repressor molecule, and it is bound to the operator. And the assumption here is that these genes are not required because they are only supposed to be expressed when lactose is available in the environment. And in this case also, glucose must not be available. It must be scarce, because when glucose is available and lactose is available, the bacteria will spend less energy utilizing glucose because it's a monosaccharide. Lactose is a disaccharide, so its preferred source of carbon or nutrients is going to be glucose. So then, this situation, you must understand, when will it happen? It will happen when in the environment there's no lactose and there's glucose, or when there's lactose and glucose is present, because the preferred source of nutrition, should I say carbon, is glucose. Now, what happens when in the environment there's no glucose, or glucose is scarce and there is lactose? This is going to change, because when lactose is available in the environment, the bacteria still needs a carbon source. So what it does is to allow for the transcription of the genes that can enable it to utilize lactose. So when lactose is in the environment, it acts as an inducer. What does that mean? Lactose can bind to the repressor molecule. When lactose binds the repressor molecule, it makes this repressor molecule change its conformation, meaning the repressor molecule can no longer bind to the operator. What happens for us to turn this gene on, and this normally will happen when in the environment glucose is scarce, lactose is available. When lactose is available, the bacteria needs to utilize this lactose. It needs to express the genes that will enable it to break down lactose so that it can utilize it for its nutrient source. To turn on those genes, lactose acts as an inducer. How is it inducing this system so that the structured genes are transcribed? The lactose is going to bind to the repressor molecule. The binding of lactose to the repressor molecule changes the conformation of the repressor molecule. And by that I mean, if the conformation of the repressor molecule was a V, and that's what enabled it to bind to the operator, it no longer is a V. So there's the binding affinity is reduced, causing it to dissociate from the operator. Now, because the operator is free, RNA polymerase can bind to the promoter and transcribe those genes. So in this case, what we can see here is that when the repressor molecule is bound, conditions in the environment must be such that these genes are not required. But when these genes are required, which could be a condition where we don't have enough glucose, but should I say glucose is scarce, but we have got lactose, the bacteria needs to survive. So it needs to turn on the genes that will enable it to utilize lactose. To do that, the lactose itself is going to act as an inducer. How does it do it? It has a high binding affinity for the repressor, changing its conformation so that it does not bind the operator. At this point, the operator is free. When RNA polymerase binds the promoter, transcription of the structural genes is going to take place. If you want a deeper understanding of this, you can look at these notes, and I'm hoping in your own free time, you can expand them. So the next thing that I want us to look at is, we've looked at regulation of the lack of operon. In negative regulation, we're saying that the repressor gene produces a molecule, and that molecule binds to operator and shuts down transcription of the structural gene. And we're saying this is not permanent. The conditions can change such that the operator can be released when glucose is scarce, and in that environment there's lactose. If glucose is scarce, there's no lactose, there is no need for those structural genes to be transcribed. What are they going to be used for? Now we want to look at the other part of the regulatory gene. We said it can produce a suppressor, it can also produce an activator. Okay, now what does an activator do? In this case, it promotes transcription, and this is going to be referred to as positive regulation. So I just want us to look at that. So in positive regulation occurs when the activator protein increases or promotes the rate of transcription. That is to say, it helps the RNA polymerase initiate transcription. I remember in prokaryotes during transcription, we looked at the sigma factor to say the sigma factor helps the RNA polymerase to load onto the promoter. Until the promoter is identified and there's unwinding and there's all that stuff, then the sigma factor falls off. Now, there are also other factors that can help transcription to take place efficiently, and one of them is to ensure that the RNA polymerase stably binds to the promoter region without falling off. What I'm trying to say here is that think of this activator molecule as a molecule, or should I say a protein, that enables stable binding of the RNA polymerase to the promoter. What that means is that you want the RNA polymerase to hold stably very quickly and transcribe your genes. So the repressor gene can synthesize a protein that can help the RNA polymerase to do that. And because it is helping the RNA polymerase to bind stably to the promoter region, it increases or improves the rate at which transcription can take place. So unlike repressors, activators are encoded by regulatory genes. Now, this one was supposed to say, well, when you think of the regulatory genes, they also produce, the activators are also synthesized by the regulatory gene. But the binding of these regulatory genes that act as activators is different. Remember in the previous slide, the repressor binds to the operator. In this case, the activator, which promotes transcription, binds near the promoter itself. Because remember, we're trying to say this activator is supposed to help the polymerase to bind to the promoter. So the sentence was to compare where the repressor binds is different. It binds to the operator and blocks it, but the activator binds to the promoter. Why? Because it helps RNA polymerase to bind to the promoter and improves transcription. Let's look at a situation of positive regulation where the repressor molecule enables the production of a protein that's going to bind to the promoter and improve the efficiency of RNA polymerase binding and transcription. So we're going to look at the catabolic activator protein, cyclic AMP, role in positive regulation. So what we see here is that we understand that when glucose is in the environment, this is the preferred source of nutrients. But then there are times when this glucose is low. Well, how does a cell know that glucose is low? And for that reason, I should, promote this system. Or should I say, how does a cell know that I've got abundant glucose? Or how does a cell know that I've got less glucose? I should look for an alternative source. So one of them is to look at an example of what is referred to as the catabolic activator protein. And this is cyclic AMP, adenosine monophosphate. So now I'm going to draw your attention to what is happening here. So when glucose is low in bacteria, the amounts of cyclic AMP increase. When glucose is available in the environment, when glucose is low, E. coli increases cyclic AMP production. Now, what does it mean? What you're trying to say is that in this environment, you don't have enough glucose. Now, for transcription to promote the transcription of these lac operant genes, we need something to improve that, to make it very quickly, to make it faster. How does that happen? The cell first of all has to recognize that I don't have enough glucose. So what happens is that when there's low glucose, cyclic AMP will be produced. Now, cyclic AMP has got a high affinity for this cap protein, and together they're going to form a complex that binds to the cap site. The cap site is a region that is found near the promoter where the cyclic AMP cap complex can bind. Now, when this complex is bound to this promoter, it allows RNA polymerase to bind stably and promote the transcription of LAC genes. Remember, we are looking at positive regulation where the transcription of these genes is promoted or enhanced. How is it enhanced? The first thing is the cell or the bacteria has to recognize I don't have enough glucose. How does it do that? It upregulates the production of cyclic AMP. In the next slide, I'm going to show you how cyclic AMP is produced. It is produced from ATP. But here, what we're trying to demonstrate is that the bacteria needs to first of all understand how does it know that I don't have enough glucose. I must switch on those genes. The first thing is to upregulate the production of cyclic AMP. When cyclic AMP is sufficient in the environment, it forms a complex with cap activator protein. This complex is going to move and bind to the cap site. The cap site is a region that is found on the promoter region that enhances the binding of RNA polymerase so that these lac operant genes are transcribed and lactose can be produced. When the glucose is high, remember the preferred source of the bacteria for carbon nutrition is glucose because it is it's a monosaccharide. So there's no need for transcription of lactose utilization genes. So when glucose is high, the cyclic AMP is low. And because cyclic AMP is low, it cannot bind to cap to form a complex and go to the cap site to promote transcription of these genes. Now, this is an example of positive regulation of the lac operant genes. Why? Because when glucose is low, cyclic AMP is upregulated, that is produced in appreciable amounts that can enable it to bind to the cap binding site and promote transcription. By this case, we're trying to say this is one of the ways that a bacteria cell knows that it needs to activate the transcription of these other genes when lactose is present. Now, because transcription is being promoted by the binding of the cyclic AMP cap complex, we say this is positive regulation of the lac operant genes. Now, I said in the previous slide, the activator molecule does not bind to the operator. The repressor molecule binds to the operator, while the activator molecule binds near the promoter. Why? Because this molecule promotes the binding of RNA polymerase and promoting its transcription of these genes. But understand, the positive regulation will only take place when in that environment there is low glucose. The cell is going to know there's low glucose. Why? Because it's going to upregulate production of cyclic AMP. The cyclic AMP will then bind to CAP, forming a complex that can bind to the near the promoter and promote RNA polymerase binding, and of course transcription. In an environment where there's high glucose, cyclic AMP is going to be low, and because of that, it cannot form a complex with CAP, because when it forms a complex with CAP, this complex moves to the CAP site and promote transcription. So when there's high glucose, the CAP does not bind to the CAP site and there's no positive regulation. Now, how does cyclic AMP get produced? Because remember, when the glucose levels are low, we have an increase in cyclic AMP. When the glucose levels are high, cyclic AMP is not, produced in appreciable amounts. This is because glucose itself can regulate the conversion of ATP into cyclic AMP by inactivating the enzyme that promotes or catalyzes this conversion. When the glucose levels are high in the environment, there's no need for us to utilize lactose. The lactose genes are repressed. How does that happen? So when glucose levels are high, glucose inhibits the catalytic activity of adenylate cyclase. Adenylate cyclase is the enzyme that catalyzes the conversion of ATP to cyclic AMP. When glucose levels are high, cyclic AMP is low. Why is it low? Because glucose is inhibiting the function of an adenylate cyclase. When the glucose levels are low, the inhibition of adenylate cyclase is lifted, and this enzyme can now catalyze the conversion of ATP to cyclic AMP. And it makes sense now that when the glucose levels are low, the cyclic AMP levels are high. And when the cyclic A levels are high, they can form a complex with CAP, and this complex is going to move to the CAP binding site near the promoter and effect positive regulation. What I'm trying to say here is that in positive regulation, you must first of all understand positive regulation in the lac operand means you're promoting the transcription of the structural genes. Now, for you to promote the transcription of the structural genes, conditions in the environment must be permissive. One of it should be such that there must be less glucose. What does less glucose mean? Less glucose means that the glucose can no longer inhibit the conversion of ATP to cyclic AMP. And because of that, the amounts of cyclic AMP are going to be high in the environment. When the amount of cyclic AMP are high in the environment, this cyclic AMP can bind to the CAP activator protein, and together the complex will bind to the promoter and promote the transcription of your structural genes, and ultimately these structural genes will be utilized for catabolism of the breakdown of lactose. For better understanding of what I've said, I've included this slide. Now, the question that we want to find out is, how did we get to know all this information? How did we get to know about the lac operand, how the operator functions, how the regulatory gene functions? Well, these um, these concepts, or should I say, these discoveries were made by two gentlemen, Jacob and Monod. These are the guys around 1951. You can imagine this is 2026. You can imagine how many years have passed. And like I always say, I'm sure when such people met, they'll be discussing, very strong, questions, challenging questions that allow one to think. Now, so Jacob and Monod, they wanted to understand how bacteria turn on genes and how they turn off genes in the utilization of different nutrients in the environment. They looked at lactose utilization, understanding that a bacteria, the preferred source of nutrients, or should I say carbon source, was glucose, for the simple reason that it's a monosaccharide. But then when glucose was not present, this bacteria could also utilize lactose. So then in an environment where you provide this bacteria with glucose and lactose, if it still retains the same potential to utilize lactose, why are the genes for lactose turned off? So they looked at such, questions, and they proposed what is referred to as the operand hypothesis. Now, I want us to move stage by stage to see how they were able to implement, or should I say, to break down the hypothesis. So we're saying that Jacob and Monod, the questions that they wanted to find out is, how do bacteria know when to produce specific enzymes? This is to say, when you grow bacteria on a culture plate, you provide it with glucose, it's going to utilize glucose. The next time, the same bacteria, when you provide it with only lactose, it can still utilize lactose. When you put it on another media where it has got both lactose and glucose, it's only going to utilize one of them first. Until that one is depleted, only then can it switch. So meaning that this bacteria, it still carries the same genetic potential to utilize lactose, only that the lactose genes are switched off. So how do these bacteria know when to produce these specific genes? How does the presence of lactose trigger the expression of lactose? That is to say, is lactose doing something to the genes in this bacteria? Could several related genes be regulated as a single unit? Because they also understand to break down lactose, it won't just be one enzyme involved. Is this bacteria turning on more than one gene to utilize lactose? And if it is, are these genes transcribed together or are they transcribed independently? So they are trying to ask all these questions, and they proposed this operant hypothesis. So we're going to go through this experiment. Excuse me. We're going to go through the experiments that were done by Jacob and Monod. So in these experiments, what they did is to, first of all, generate a diploid bacteria. You know, a diploid means it has got, in this case, it should have two copies for genes that are involved in your lactose utilization. So they obviously need a wild type. The wild type is the one where these genes that are involved in lactose utilization are functioning. And the mutant strains, these are strains where some of these genes that are involved in lactose utilization are turned off. And this is to say, for example, you want to find out if a given fertilizer improves your maize yields. So you're going to have one treatment should have no fertilizer, and another treatment should have fertilizer. So when you think of what Jacob and Monod are doing, they needed to have a control. The control is a wild type, the natural system where the structural genes are functional. And then the mutant strains, these are the ones where now you start switching off some of these genes that you suspect are involved in lactose utilization. And also they measured the expression of beta-galactosidase, the LACZ. Remember, beta-galactosidase from that one of the slides, this is the enzyme that is going to be used to break down lactose into glucose and galactose. And also they constructed diploids. So in this case, a diploid, in reference to the lac operon, this is a bacteria that was carrying two copies of these structural genes. What is referred to here as diploid is, in the experiment, they worked with bacteria, either the wild type or the mutant, that was carrying two copies of the lac utilization genes. So if you look at the LAC-Y, LAC-Z, and LAC-A, it means that bacteria carried two copies of the LAC-A, two copies of LAC-Z, and two copies of the LAC-Y. And then, of course, they mapped these mutations to see how they were affecting gene regulation. Let me use a slide to make this simple for me to explain. So right now we understand what the operant structure looks like. What you're shown here, if I stand at this point, you have got your regulatory gene. In this case, it is designated as a LAC-I gene, like lactose inducer. And then you've got the promoter. This promoter, this is the one where RNA polymerase binds to transcribe the structural genes. Then we also have an operator region. We said the operator, this is where your repressor molecule binds. And of course, you've got your different structural genes. So now, what they wanted to understand is, how are these lactose genes turned off? So in their first analysis, or their first observation, is that genes for lactose utilizations are switched off when lactose is not in the system, okay, in the environment of the bacteria. And this is because when lactose was not present or absent, okay, the regulatory gene will produce the regulatory protein. This regulatory protein will bind to the operator region. You can see that the repressor molecule has got a certain shape that fits well into the operator region. When this repressor molecule binds to the operator, the transcription of these structural genes is off. And this only happened when this environment did not have lactose, or when this environment, even if there was lactose, glucose was present. One of the observations or the experiments led to the discovery that when lactose is absent, or when lactose is present but glucose is available, the regulatory gene will produce a repressor molecule. This repressor molecule will bind to the operator and block transcription. This is what we refer to as negative regulation of gene expression. Now, what will happen when this repressor molecule is removed? I'm going to skip these slides because these are just notes that you can go and read. I want to go to the picture that will show us what will happen when the conditions in the environment change. That is to say, in the environment when we have lactose, glucose is scarce. What happens to the lac operon? Is the repressor molecule still going to remain bound to the operator? So what happens in that system is that repression is lifted. So the regulatory gene will still produce the regulatory gene. But then the regulatory gene does not bind to the operator because in this environment, lactose is present and lactose binds to the repressor molecule. The binding of lactose to the repressor molecule causes a conformational change that does not allow the repressor molecule to bind with affinity to the operator. Because the operator is free, the transcription of these genes takes place and lactose in the environment can be utilized as a nutrient source. Now, I want us to pay attention to where I'm hovering my cursor. The repressor protein is not binding to lactose in its form as lactose. It is binding to a form of lactose that is referred to as allolactose. This is an isomer of lactose, and the difference here is just how the lactose has got a beta 1,4 glycosidic bond. This one has a beta 1,6 glycosidic bonds. And at this point, from our background of monosaccharides, we understand what an alpha 1,4 and 1,6 glycosidic bond means. So what we're trying to say here is that for this repressor molecule to unblock the operator, we need lactose to be present in the environment. But what form of lactose is binding to this operator? It is the allolactose. Allolactose then acts as an inducer. What this inducer does is to inactivate or stop the repressor molecule from binding to the operator. And when this happens, the transcription of these lac genes is going to take place and lactose can be utilized in that environment. I just want us to look at the lactose itself to understand the allolactose is a form of lactose that binds to the repressor protein, changing its conformation so that it does not bind to the operator. The conversion of lactose to allolactose is also catalyzed by beta-galactosidase. So what this means is that some of the lac genes, despite the repression by the operator, because remember, initially we had the operator that was blocked, meaning that the structural genes are not being expressed. But then it's important to understand that for us to have lactose being converted to allolactose, beta-galactosidase, which is among the structural genes, must be present. What this means is that despite the operator binding, despite the repressor molecule binding to the operator region, there's basal expression of the lac genes, meaning there's a leak. There's not sufficient amount to allow the bacteria to utilize lactose, but some of these enzymes are being produced, or should I say transcribed. This transcription is referred to as basal transcription. Basal transcription meaning that the transcription is taking place minimally, or there's a leak. The pressure is not 100%. Okay, because, and this is important because when lactose is available, the lactose has to be converted to allolactose by some of the enzymes that are in the structural genes. In fact, this same lactose has to enter the cell, so meaning the lactose permease also has to be transcribed. So we're trying to say that Allolactose is made possible because of transcriptional leak. When we say the operator is blocked by the repressor protein, this repression is not 100%, meaning to say there's zero transcription of these lac operand genes. There is minimal transcription, and that is what we're referring to as basal expression or gene expression. There's a leak in gene expression. The enzyme that is converting lactose to allolactose is beta-galactosidase, meaning that beta-galactosidase has two functions. It can convert lactose to allolactose so that this can function as an inducer. It can also function in the catabolism of lactose, breaking it down into its monomer units. So when you look at the structural genes, beta-galactosidase is doing two things. It can allow for genes to be turned on. Okay, it can allow for the conversion of lactose into allolactose. At the same time, it can allow for the breakdown of lactose into its monomer units. For more information on that explanation, you can look at this slide. Now, I know I don't have a lot of time with me. I'm not too sure whether I can have just 10 minutes of your time just to explain how the operator and the inducer gene work. Now, some of the discoveries of Jacob and Monod experiment was to understand how the operator works. To start with, we are saying that what they created were diploid cells, diploid bacteria cells. By diploid, you mean they're carrying two copies of the same gene. So they wanted to find out, how does the operator work? The operator is this region that binds the repressor molecule. When the repressor is synthesized, this operator binds to the repressor molecule. Now, let's pay attention to the first gene, okay, the first chromosome, okay? We have got our repressor gene, then we have our promoter for the structural genes, and our operator, and then we've got LAC-Z and LAC-Y. Now, when you have a plus on the gene, as you can see, this is I plus, P plus, O plus, Z plus, and plus. This means this is wild type. There is no mutation. All these genes are functional. What you're shown here is that when you see any of those genes, there is a plus, meaning that these genes are wild type. They're going to produce functional RNA and functional proteins. Now, let's now go to this other chromosome. You also have the LAC-I gene. For the repressor gene, it is plus, meaning that it is functional. The promoter here is also functional, but the operator there, you've got a C. When it is designated as a C or a negative sign, it means that this is a mutation. This point here cannot bind the repressor molecule. What this means, when you have a mutation here or a mutant portion at this position designated as a OC or sometimes O minus, what it means is that this point here is mutated, it's distorted. It can no longer bind the repressor molecule, even if it is in this environment. Again, your LAC-Z is positive, meaning this point is functional like the wild type, and this point is also functional. So now, they wanted to understand, when this repressor molecule is synthesized in this environment, how does it affect the transcription of those genes? Does it mean that when it is present in the environment, it's going to repress both the genes on this chromosome that has got a mutant operator, or it's only going to affect those that have got a wild type operator? What they noticed is that when the operator is functional, that is the only condition under which it can bind the operator, meaning that this operator can bind the repressor, and the transcription of these genes is going to be blocked. But let's move to this chromosome. Regardless of the repressor molecule being in this environment, because the operator region is mutated, it cannot bind the repressor. These genes, the transcription of these genes cannot be blocked. What this means is that this operator is functioning in cis. By cis means that it can only affect its function, its its ability to repress can only work on the genes that are found on its own DNA or its own chromosome. If you look at this operator region, it's functioning in cis because when it is mutated, these genes cannot be repressed. So what they discovered is that the operator functions in cis. When this operator is not functioning, it is not affecting the regulation of genes on this other chromosome. Or indeed, when this operator is functioning, it can bind the repressor molecule. It cannot block the transcription of these genes. So cis means it can only affect the genes on this chromosome. If it were in trans, it would mean that when the repressor binds to this molecule, to this operator region, it was also supposed to block the transcription of these genes. But because it is functioning in cis and not in trans, they said that, or they concluded that the operator is a cis-acting regulatory element. By cis, it means when the operator binds the repressor, it can only affect the transcription, in this case, block the transcription of its own structured genes. It can't affect the transcription of the other genes where it is not present, like what you can see here. And also, the take-home message from this slide is that when you look at the repressor molecule, if it can't bind the operator, it means these genes are going to be constitutively expressed. They're going to be turned on every time because there's nothing that is blocking their transcription. Remember, RNA polymerase will bind because the promoter is working. The repressor molecule here is just acting as a switch, and because the switch is not working, these genes will be continuously transcribed. There's nothing that's going to block transcription. So in this environment, I want us to start following from this slide that this operator functions in cis, because I'll need us to use this information to read out combinations of mutations. Okay, if you want to understand more, I've added this slide for your notes. Now, what about the repressor molecule? How is the LAC-I gene functioning? The LAC-I gene here is the repressor gene. When it produces its product, does this product function in cis, meaning it's only affecting the genes on its own chromosome? It can affect even the genes on the other chromosome. Remember, they are working with a diploid system where you've got two copies of the same operand. So let's look at the first chromosome or the first DNA. You have your LAC-I gene. Like I said, this is a regulatory gene. You can see it is I minus. Minus, I said, this is a gene that is not going to give us a functional product, or in this case, it is mutated, meaning we're not going to get our repressor molecule from here. It's a mutant. But when you go to the promoter is active. What this means, it can bind RNA polymerase. The operator is also working, meaning that if the repressor molecule is in the environment, it can bind it, and the structural genes are also working. What about this other gene? This other gene, we can see that the LAC-I gene, the repressor gene, is functional, meaning it can produce an active repressor. Now, this repressor, they wanted to find out, can it bind to this operator and that operator? Remember, both of these operators on these genes are functional. You can see this is O plus and this is O plus, meaning that their conformation can allow for binding of the repressor. And they found that when the repressor molecule is produced, regardless of where it came from, either of the genes, it can elicit inhibition or blocking of transcription on both genes. What this means is that the LAC gene or the repressor molecule acts in trans. It can affect the repression of its own gene where it was produced. It can also affect the repression of the other genes on the other chromosome. So now we need to distinguish the operator, which only functions in cis, and the repressor molecule, diffusible element. While the operator region is not a diffusible element, it's part of the DNA itself, so it can't move about. So maybe this is why we're saying that the repressor is acting in trans, because it can diffuse into the system and bind to any operator that is functional. While the operator functions in cis because it's an intrinsic feature of that DNA standing in only one position, and therefore can only affect blocking or repression of the genes where it is located. For you to have a better understanding, they are not for you here. This is our last slide. So now, I'm going to end here, but I want us to look at these phenotypes, like I'm trying to say. This requires us to understand how the repressor the repressor gene is functioning. Remember, we said it functions in trans, but the operator functions in cis. So think of an organism that is a diploid. A diploid means it has got two copies of the same gene. This is one copy of that operon, and this is the other copy. And what you're given here is a condition where lactose is present, and you're being asked, if lactose is present, will beta-galactosidase be present? Will it be transcribed, or the lactose permease? Under what conditions? This genetic combination. And also, you can be given another condition. When lactose is absent, are you going to have any LAC-Z and the LAC-Y? Under these conditions, let's pick A. Okay, let's look at what A is saying. So we have this gene, or the LAC-I gene is mutated. And we said that when we have this condition, we're not going to get our repressor protein, right? Now, let's look at the operator. The operator region is working. It is positive, meaning that if in this cell there's a repressor molecule, it can bind to it. The LAC-Z, which is going to give us beta-galactosidase, is negative, meaning that this gene is not functional. Meaning that even if there was transcription of these genes, we're not going to get any gene product from here. What about the lactose permease, the LAC-Y? The LAC-Y is plus. What this means is that this gene here, this gene copy is functional. Transcription will give us mRNA, and we can get a protein after translation. This is a combination of one gene. Non-functional is very functional, meaning it can bind the repressor molecule if it is present. The LAC-Z is not functional, but the LAC-Y, we can get it because it is functional. Let's look at the other copy. Here we have got a functional repressor gene, meaning that it can produce the repressor protein. On this copy, in this same bacteria, we have got a functional LAC-I. A functional LAC-I because we can say it is I plus. I plus means that this position or the regulatory gene is going to give us a repressor protein. What about the copy of the gene copy of the operator? It is OC, meaning that this is a mutation. Meaning that even if this gene copy can produce a repressor protein, it cannot bind to its own operator. If it cannot bind to that operator, what this means is that we're going to have, and the LAC-Z here, you can see it is positive. So meaning on this copy, it is functional, but the LAC-Y is non-functional. Now, interpretation of what I'm saying sometimes is a bit complex, and you need to follow through and look at this combination with some form of patience, okay, to understand what is functional on this gene copy and what is functional on this gene copy. So now, if you have followed what I was trying to explain about these two combinations of gene copies, let's look at a condition where lactose is present. And the assumption here is that glucose is not present, okay? Glucose is not present because remember these guys wanted to find out how is lactose utilized. So maybe they take out glucose. So lactose is present. So if lactose is present, are we going to get enzymes for its utilization? That's what they're trying to ask you here. So now, we know that here, the repressor molecule won't be synthesized, but the repressor molecule here is going to be synthesized. And when the repressor molecule is synthesized, it functions in trans because it's a diffusible molecule. So this protein, it can't bind to its own operator, but it can bind to this operator. And when it binds to this operator, it means that it's going to block the transcription, not of the LAC-Z because this guy is mutated, but it's going to block the transcription of the LAC-Y. Let me say that again. So Let's look at the condition when we have lactose that is present. Are we going to get the genes for lactose utilization? So we know we're going to get, in this system, we're going to get a repressor molecule, which is a the repressor protein, because the lac gene is functional. When it's functional, we know it is functioning in trans. It's going to block the transcription of these genes because it can bind the operator. So meaning that when lactose is present, sorry, let me just take that out. Okay, it can bind to this molecule. But remember, when lactose is present, what does lactose do? It is converted to allolactose, isn't it? When it's converted to allolactose, the repressor molecule has got a high affinity for allolactose compared to the operator. So meaning that from this gene copy, we're going to get repression and production of the LAC-Y gene, but not the Z. What about this end here? Now, remember, the operator region is not functional. So meaning that while in this system, we have got the repressor molecule, whether lactose is present or not, these genes here cannot be repressed or blocked because transcription, the polymerase will bind the promoter and synthesize the structural genes. Now, while we can't get a functional LAC-Z here, we can get a functional LAC-Z because this protein here is functional, or this gene here is functional. But we can't get a LAC-Y because this gene is not functional. So when lactose is present, we're going to get beta galactosidase and beta pepamase, lactose pepamase. Where did the beta pepamase come from? It came from the second gene copy and not the first one, because the first one, this point is not functional. What about the lactose pepamase? Where did it come from? It came not from this gene copy, but it came from that gene copy. When lactose is present, we understand it's going to be converted to allolactose, and the repressor molecule, even though it has been synthesized, it cannot bind. But what happens when lactose is absent? When lactose is absent, it means we don't need the lactose utilization genes. But are we going to have them in this system here if lactose is absent, given this combination? We understand each of these combinations and what they mean. So when lactose is not present, what it means is that this functional repressor molecule is going to synthesize lactose. Sorry, this functional repressor gene is going to synthesize the repressor protein. That repressor molecule cannot bind to its own operator. But because we know it's a diffusible molecule and it works in trans, it can diffuse and bind to this operator. And because lactose is not present, it will continue binding to the operator. Now, when it binds to the operator, it cannot block LAC-Z because LAC-Z is not functional at this point, but it's going to block LAC-Y. So meaning in this environment, we won't get any lactose pepamase. Why didn't we get lactose pepamase from this position? Because in this gene copy, this lactose is mutated. Now, let's move on to this other copy of the gene. In this environment, we have our LAC gene or the LAC protein, but the LAC protein can't bind to this operator. Because this operator is free, it can allow for RNA polymerase to move and synthesize the structural gene. But we need to be careful. Only the LAC-Z here is function, but the LAC-Y is not function. So in an environment where the operator is mutated or is non-functional, there's going to be continuous expression of the structural genes. But we know only the LAC-Z is going to be present. So yes, even if lactose is not present, this bacteria is going to spend or waste energy producing beta galactosidase because the point where it is supposed to be suppressed is mutated. I'm going to end here, and I'm hoping that you can go on to understand each one of these. I've also included a slide for you to look at. So up to this point, this is where the lecture will end. But like I'm trying to say, these combinations of LAC-Z, LAC-Y, and how the repressor and the operator function require you to be a bit patient, to understand what each one of them means in the presence of lactose and in the absence of lactose. And yeah, if you need help, you can always reach out and they can run you through. In fact, this is how the questions might look like in your test. Okay, you'll be required to interpret things such as this. Okay, so this one is a single genotype, meaning there's only one copy. This one is a diploid because it has got this gene copy and this gene copy, meaning that at each position you've got a functional regulatory gene being produced and a non-function. Because it is diffusible, it can affect both of these. So in the absence of glucose, lactose, and the presence of lactose, they're going to behave differently. Guys, have a good day ahead and enjoy the weekend on Monday. Unless there are any questions, we can call it a day. Okay, there are no questions. Enjoy the rest of the day.